Hello, grade 9. Hope you're doing well. Today, we're going to do a revision of Activity 1, Transmission of Genetic Information, mitosis, and, of course, solve together some exercise. So, what is mitosis? Let's again define mitosis. Mitosis is the process of cell division which results in the production of two daughter cells from a single mother cell. And as you know, the daughter cells are identical to one another and to the original mother cell. But can any cell start division at any time? Of course not. First, nerve and muscle cells, they don't divide. Second, each cell must first undergo a growth phase called interphase. So, before any division, a cell grows up from a daughter cell into a mother cell by interphase. Then, after interphase, the cell is ready to divide by mitosis. That, if you remember, it consists of 4 phases: prophase, metaphase, anaphase, telophase. So, interphase plus mitosis, they make one cell cycle. Let's take an example of mitosis of an animal cell to do a recap together. A daughter cell, or it could be a zygote with 2 N chromosomes. Every chromosome of one chromatid, and you see this chromosome is long and thin with 1 centromere. This daughter cell undergoes interphase during which there is duplication of chromatids and becomes a mother cell, always with 2 N chromosomes. But this time, the chromosome is of 2 chromatids joined together by a centromere. Remember that during interphase, chromosomes are always long and thin, and we call them chromatin. Now, once the cell is adult, mature, a mother cell, it's ready to start a cell division, which is mitosis. First of all, it starts with prophase. As you see, the number of chromosomes always 2N, but the chromosomes that were long and thin, they condense to become short and thick. Also in metaphase, the cell keeps 2N as number of chromosomes with short and thick chromosomes of 2 chromatids each. However, in the third phase of anaphase, with 2N chromosomes at every pole of the cell, but every chromosome has now 1 chromatid. Finally, the cell divides in the last phase, telophase, into 2 daughter cells, each with 2 N chromosomes, but unlike the mother cell in prophase, the cell has long and thin chromosomes of 1 chromatid. And this is what we call decondensation. It's when the short and thick chromosome becomes long and thin. So, condensation occurs when the cell passes from interphase into prophase. The chromosome of 2 chromatids, long and thin, becomes of 2 chromatids always, but short and thick. While decondensation happens in telophase, and the cell that is short, that has chromosomes short and thick of 1 chromatid, has now also 1 chromatid, but long and thin. Let's take the example of mitosis of an animal cell with 2N equals 6 chromosomes. So, how many pairs of chromosomes does this cell have? If it has 6 chromosomes, then this means that it has 3 pairs of chromosomes. Let's see. The cell with 2n equals 6 chromosomes undergoes a first growth phase called interphase. The cell has in the cytoplasm a centrosome made up of 2 centrioles, a nucleolus inside the nucleus. The nuclear membrane is very visible, the plasma membrane, and inside the nucleus we have the chromatin, chromosomes that are long and thin, filamentous, and that are not visible and we cannot count them. Remember, in interphase, the chromosomes double. So every chromosome at the end of interphase has 2 chromatids. But always we cannot see them during interphase. This cell undergoes prophase, the first phase of mitosis. First of all, the centrosome divides into 2 pairs of centrioles, each pair becoming an aster, joined together by the achromatic spindle or spindle fibers. The nuclear membrane starts disappearing progressively, and the chromosomes are now visible. We can count them. This is, for example, 1 chromosome of 2 sister chromatids. They are not very, very short. They become short more and more in the next phase or meta phase. So, always as you see, the cell in interphase has 2n equals 6 chromosomes, but they are long, thin, called chromatin. In prophase, the number of chromosomes is always constant, 2n equals 6, but they become shorter, thicker. Each chromosome has 2 sister chromatids. Next, the following phase is metaphase. The 6 chromosomes line up in the center of the cell, forming the equatorial plate or the metaphase plate. Next, anaphase. Every chromosome of 2 chromatids of these chromosomes divides into 2 chromosomes. Now every chromosome has only 1 chromatid. So we obtain in the upper pole 2n equals 6 chromosomes, in the lower pole 2n equals 6 chromosomes, every pole with 1 chromatid. Then the cell reaches its last phase of division, which is telophase, during which cytokinesis takes place or division of the cytoplasm. You see clearly the nuclear membrane forms in every cell, of course, after cytokinesis or cleavage furrow, which is the constriction of the cell into 2 daughter cells. And in every nucleus, we have again 2n equals 6 chromosomes, but this time chromosomes are long and thin. So every cell of these cells has always 2n equals 6 chromosomes. As you see, the number of chromosomes is conserved during mitosis. Let's start with the first application. This is a drawing of a cell during mitosis. First question. Label the structures A to D. What is A? What is B? What is C? And what is D? Dear students, try to answer, please, these questions. A corresponds to the aster. B corresponds to the chromosome of 2 chromatids. C is the cytoplasm of the cell. And D is the plasma membrane. Second question. Identify this phase. What does this phase correspond to? Yes, this phase corresponds to prophase. When the question is to identify, You need always to justify your answer. So, how do we know that this is prophase? Since the nuclear membrane is disappearing progressively and the chromosomes are visible inside the nucleus, thick, can be counted, made up of 2 chromatids each, then this is prophase. Third question, indicate the number of chromosomes in this cell. Let's count them. 1, 2, 3, 4. You can say 2 pairs since every 2 chromosomes are homologous. You can see orange and brown. They form 1 pair of homologous chromosomes as well as the 2 other chromosomes. So the number of chromosomes is to n equal 4 or 2 pairs. Question number 4. Draw this cell during the phase that follows this phase in the figure here. What is the next phase? If this phase is prophase, then the next phase would be metaphase. This is metaphase, and the chromosomes are aligned at the equator of the cell, forming the equatorial plate. Another application. The diagram below represents the stages of a cellular process. A, B, C, and D. First question, name this process. What do you think? What is this process represented in its 4 stages? Of course, the process is mitosis. Second question, arrange these phases in chronological order. What's the order? C, then B, then D, and finally A. Why? Let's see. Question 3. Identify each phase. First phase, C. This is prophase. Why? Because the nuclear membrane is disappearing progressively and chromosomes are thick and visible. B is metaphase. Look at B. Whenever you see chromosomes in the center of the cell forming the equatorial plate, then this is metaphase. D, it's anaphase. Since there is polar ascension, each chromosome of 1 chromatid moves to 1 pole of the cell. So you see polar ascension, chromosomes gathered at every pole. Finally, A, this is telophase, since the cell shows a constriction and is dividing into 2 daughter cells. Whenever you see a constriction, then this cell is telophase. Indicate the number of chromosomes of the cell. So let's count them. 1, 2, 3, 4, 5, and 6. Then the number of chromosomes is 6, or you can say 3 pairs of homologous chromosomes. Dear grade 9 students, this is a homework, please, for you, and this homework is due tomorrow, Thursday. Thank you for your attention. Take care of yourselves. See you later.